We work on DNA methylation. DNA methylation is one of the epigenetic alterations that is known in the human cell. DNA methylation, histone modifications, chromatin positioning are all considered as epigenetic alterations, and altogether they are important in regulating gene expression. And we are interested in cancer, in leukemias, in chronic lymphocytic leukemia in particular here. And our question is, how do epigenetic alterations contribute to the leukemic phenotype? And we have known for many years that DNA methylation patterns can change, and that this has an influence on the gene expression of cancer genes that contribute to the leukemic phenotype. It's very important because we know that the B cells from which CLLs originate already undergo a massive epigenetic programming. So about 30% of the whole genome is changing on the DNA methylation level, and depending on the timing and the maturation stage in which a B cell is, when it becomes transformed to a CLL, it carries a different epigenome. And this epigenome consists in the background and regulates gene expression, and becomes very important in understanding how a patient will respond to therapy. This is determined by gene expression patterns, for example, mediated and programmed by epigenetic alterations.